Hey everyone, Graham Buckner back here. We're going to do the final activity, which is activity 4, and we're going to talk about the domain eukarya. Okay, so let me fast forward to where we were. Sorry about that. I'm going to go ahead and minimize my video. Okay, a few more seconds. Okay, domain eukarya. All right, this is going to be our 3 kingdoms. Underneath the domain eukarya is going to be our protists, animals, and fungi. All right, these are cells are large and they're going to have subcellular structures. Okay, and they're also going to organize tasks into those subcellular structures. So think about we have chloroplasts inside eukaryotic cells, which will do photosynthesis. They also have specialized flagella in a 9-2 structure, and again, we'll talk about that. And these guys can be single-celled, colonial, or multicellular. So here's our domain eukarya. Okay, and this is our eukaryotes. So they're all going to share the characteristics of that domain. And in this domain, we've got, again, these different groups here, animals, fungi, and plants. If we look at its phospholipid bilayer, we can see that here. And it's we've got polar heads, phospholipid molecules, and non-polar tails in the middle. And essentially, it creates a really good barrier between the external environment and the internal environment. We can also see a lot of embedded proteins to transfer things across that membrane. Eukaryotes are going to possess a flagella. Okay, it's very common in single-celled eukaryotes. In multicellular eukaryotes, it's going to be present in specialized cells or for at least part of the life cycle. So, if you think of a human, we don't have a big flagella stuck to us to move us around, but we do have flagellated sperm. Some things will have flagellated larvae, and we'll also see, like sponges, have specialized feeding cells which use flagella. Okay, each flagella is composed of protein rods. Okay, not a singular helical rod like our prokaryotes, but we have multiple rods. Okay, what we actually have are 9 sets of microtubules around the outside, so you can count those, and then 2 sets in the middle. Okay, these sets are also called dimers. So 9 are dimers around the outside with 2 in the middle, and that's why we refer to it as a 9+2 structure. For the movement, it's called a wagging motion because essentially those proteins, microtubules on the outsides, are going to lengthen. We're going to shorten the side, lengthen in the other, and then shorten the other one. Okay, and what that does over and over again, if you kind of watch me for a sec, it creates a back and forth motion, and eventually that creates a big wagging motion back and forth. Okay, so if we think about how they're moving around, they've got a wagging motion as opposed to a hook and motor propeller type motion in our prokaryotes. The flagella is also covered in the plasma membrane in eukaryotes, which again is different than our prokaryotes. So here we have our plasma membrane, and it actually covers the entire flagella. We're going to talk about protists this week. We'll talk about the other domains for the rest of the semester within eukarya, with the other kingdoms within eukarya, excuse me. But protists are kind of a mixed bag. So we have they're mostly unicellular microorganisms. They don't form tissues. They can have characteristics of plants, animals, fungi, or combinations of those. So it's a really wild group, and they're really common in water sources or at least very moist environments. If we look at the lineages of protists, the first thing we want to note again is that all eukaryotes are protists except for fungi, animals, and plants. Right? Protists are typically unicellular, but as with biology, there's always exceptions. So there are multicellular protists like the macroalgae, and typically we'll classify them as in uniconta or biconta, having 1 flagella una, or bi 2 flagella. Okay, so that's a way we can group them together. But again, within another exception to the rule, members of Staminophylla can have numerous small flagella, and we kind of call them cilia, and so we're not going to classify them as uniconta or biconta. Protists can be plant-like, animal-like, or both. So if it's plant-like, it's not going to be moving around. It'll be green. It's going to have chloroplasts with chlorophyll and doing photosynthesis. If it's not colored and it's moving around, that's a pretty good indication that it's an animal-like protist. And if it's green and it's moving around, then it's going to be plant and animal-like. Okay, so just realize that there's multiple combinations possible out there. For looking at the different lineages, again, focus on the red box for defining characteristics. Use those characteristics to help you remember which group is which. Okay, just because it's not in the red box doesn't mean it's not important. If it helps you remember it, by all means, use it. Okay, and keep in mind some of the other things we just talked about, whether it has one flagella or two, and being in uniconta or biconta. Okay, something that we would do in lab typically would go to a pond, grab a sample of water, bring it back, and let you guys explore around in it. This year, because we're online, we have pre-recorded little videos and pictures of what you would see in a simple pond sample. Okay, these can have prokaryotes, eukaryotes. They may have bacteria and archaea. They will have bacteria, or possibly archaea and protists. And this is a good way to test your ability to distinguish between cell types and domains. And we're also going to take this opportunity to work on your drawing skills. All right. So kind of treat this as a prep for lab practical, where we could put something on there and ask you to identify it. Okay. So when we're looking under a microscope, we want to note what magnification we are at. We're at 40x. All right. So we have a little protist here. All right. And so if we were to ask you if this is prokaryote or eukaryote, I kind of just gave it away. But since it's large and we can kind of see some distinguishable internal features, we can see something's going on in there. This is most like a eukaryote, and since it's green, it's going to be at least plant-like. If it was moving around, it could be animal-like and plant-like. If we're looking at these, again, super tiny under the 4x objective lens, if you had to guess, which would it be? It's going to be a prokaryote, okay, really small, can't see anything, most likely a bacterium. Again, pond water is not an extreme environment, so it's probably not an archaea, so we're pretty confident calling this a bacterium. All right, and what about this? Okay, so if you look at that, you go, well, that's big, so it's definitely going to not be a prokaryote. But if we look in here, we really can't see any internal features, and it turns out that this is an air bubble. Okay, so we always point this out. Just so you make sure that you draw something that's actually alive. Okay, and the air bubbles can be tricky when you're looking through those. All right. Proper technique for making a wet mount is just something every biologist should know how to do. So we're just going to put a little drop of water on the slide, and we have a little cover slip, and we're going to put it just barely on the edge here and gently set it down at an angle. And what that does is it keeps a nice even spread of the water, and it keeps things from bubbling out. So if you just plopped it down on top, you'd probably spray water everywhere, and you could lose some cool protist or bacteria you could lose it, and it could not be seen under a microscope. Okay. If we had a little excess water here, we can just simply use a Kim wipe to wipe it up. Okay. I want to correct something I said in the first video. In the past, we've had you guys use a formula to calculate the field of view at the 40x. This year, we're not doing that. It should be listed in your worksheet. So don't worry about the formula that I was talking about. Just ignore it. We already gave you those measurements. Okay. So we're going to talk about estimating cell size now, because if we think back to the difference between prokaryotes and eukaryotes, a big one is the size of it. Okay. And if we can estimate the cell size, we've got a pretty good idea whether or not it's a prokaryote or a eukaryote. All right. So how would we go about doing this? Well, the first thing we note is that we know this distance. So that's going to be helpful. Okay. What we're actually going to do is use that. And we're also going to determine how many of these little cells right here could fit across this screen right here. So 1, 2, 3, 4, 5, 6, 7, 8 cells can fit across that field of view. Okay. Note that we're calling this a bacterial cell, but if we were under 10x, it would not be that big. This is just an example. Okay. I don't want you guys to get confused by that. Okay. And we could simply just measure this field of view. Here it's going to be 1.75. All right. And we estimated that 8 cells could fit across there. Okay. And we're simply just going to divide. Okay. So if we divide the field of view by the number of cells, we're going to get 0.22 millimeters per cell, which is essentially telling us that we are at 0.2 millimeters for the size of a cell. Okay, we want to convert that from millimeters to micrometers. I don't think that's showing up on there. I can't see it on my screen. So we want to convert from millimeters to micrometers, because that's the measurements we're thinking about for our cell size for prokaryote or eukaryote. And to do that, we would say, well, do your train track. So you got 0.22 millimeters at the top. Okay, and you would say in 1 millimeter, there are 1,000 micrometers. And so you could do that math to figure out the size in micrometers of our cell. Okay, that's a little confusing, no problem. You know, a lot of people tend to struggle with math, but that's okay. Ask your lab instructor, email me, we can always help out with that. Okay, so that is the end of the activities for exercise 2, and hope it was good for you guys. I enjoyed it. Make sure you get through all the activities, and if you have any questions about anything, again, just reach out to your instructor or me, and we'll be more than happy to help you out.